Hi, my name is Runzhi Zhang. I'm a PhD student in the Department of Biostatistics at the University of Florida. Today I will talk about unraveling T cell responses for long-term protection of SARS-CoV-2 infection. So as we know, almost all the countries around the world suffered from the COVID-19. Until now, there are about 189 million confirmed cases, including about 4 million deaths around the world. And currently, many different types of vaccines have been developed, and improving antibody responses to the spike protein is the current main research direction of vaccine development. Since CD4 T cells play a crucial role in regulating antiviral immune responses through the secretion of a specific cytokine, understanding the mechanism of T cell responses to SARS-CoV-2 can provide more knowledge on vaccine design. So in this study, two datasets were used. The primary dataset is the single-cell RNA data of CD4 T cells from blood samples, which contains cells from both COVID-19 patients and healthy donors. And our package, Ferret, was used for normalization and scaling for the single-cell dataset. Also, we included a bulk RNA-seq data of whole blood samples, which contains the information of time from COVID-19 symptom onset. We used this dataset to trace the changes of gene expression longitudinally. Firstly, we did the differential expression analysis, aka DE analysis, for the single-cell dataset to obtain the differential expression genes using the R package MAST. Since there were much more cells in COVID-19 samples, cells from COVID-19 patients were downsampled first and then combined with cells from healthy donors to be used as the balanced dataset. So we generated 100 balanced datasets, and the genes being identified as DE genes in all of the balanced datasets will be regarded as our final DE genes. So once we obtain the DE genes, we used these DE genes to build the networks for healthy donors and COVID-19 patients respectively based on PIDC algorithms. So module detection and hub gene detection were then implemented for each network, and permutation test was used to evaluate the differential connectivity of a single gene between two networks. For example, if gene A connects to 10 genes in the healthy network but only connects to one gene in the COVID-19 networks, then gene A might be the differential connectivity gene. And in addition, random forest was used to do the classification of the COVID-19 group and the healthy group based on the 100 balanced datasets using all the genes included. An important score was obtained from the random forest, so the most important genes were with the highest important scores. And after DE analysis, we built the networks based on the DE analysis. So age in the network indicates the connectivity between the genes, and the red nodes indicate the hub genes. As we can see from figure 1, the genes with the same module are connected to each other. And the modules of the COVID-19 network have changed compared to the modules of the healthy network. And genes in module 1 of the COVID-19 network are almost the same as gene in module 1 and 2 of the healthy network. And we've also found that all the significant differentially connected hub genes are involved in the inflammatory and immune regulatory processes, indicating the possible changes related to these functions between COVID-19 and healthy subjects. And in addition to the networks, we have done function annotation to the DE genes also. And as seen from the figure 2, we found that different modules were annotated to different functions. However, it seems that Module 2 of the healthy network and Module 1 of the COVID-19 network shared a similar functions, both related to viral transcription. Also, by comparing the top 100 important genes obtained from the random forest and the DE genes included in the network, we found that 51 of the important genes were not selected for the network. These genes were related to the myeloid cell differentiation according to the function annotation. So to give you a summary, several analytical methods including DE genes, network analysis, and classification have been implemented to explore the mechanism of gene expression of CD4 T cells related to COVID-19. The results based on these approaches might improve the insight of gene expression associated with CD4 T cells, expand our understanding of COVID-19, and help for future vaccine development for long-term protection. And that's all. Thanks for listening.